Hi, I'm Camille, and this is producing clones. What is cloning? There's two types. There's animal cloning, which is creating an organism that has the exact same genetic copy. It means no difference at all. There's gene cloning, which is isolating a single strand of DNA and making an exact copy of it, much like DNA replication. And it's important that you know that it is exactly the same DNA. Uses of cloning. There's lots of uses. There's medical uses. We can use clone animal models to create vaccinations. We can clone stem cells to advance stem cell research. And farming, which is genetically modifying animals and plants to create pharmaceuticals. We can also revive endangered and extinct species. Here's a picture of a mountain goat they revived in 2001, but unfortunately died. You could reproduce diseased pets, like say if my cat dies, and I could just bring it to a cloning laboratory and have it cloned and have my cat back. Cloning humans is controversial, but it will be covered in another video. It could help infertile couples and replace deceased infants that die early. And you can clone it and you'll have your baby again. How does cloning work? There's most important is somatic cell nuclear transfer, and to understand this you have to break it down into parts. There's somatic cell, which is anything but a sex cell, a nuclear with nucleus, and transfer. So basically it's the transfer of the nucleus of a somatic cell. There's two videos, enucleation and transfer. And in nucleation, the person basically sucks up the nucleus of this egg cell, sex cell, and then here we transfer the somatic cell nucleus into an empty egg cell. And the second part is artificial embryo twinning, which is basically how natural twins are formed, the division of an egg of a forming embryo. It's done in a petri dish in a laboratory, and it's done manually separated. The big picture, basically a natural reproduction. There's mother and father, sperm and egg. And it creates an embryo and you have a baby. And somatic cell nuclear transport, basically they take the somatic cell in a petri dish, you culture it, then you get the egg cell donor, take out the nucleus of the egg cell, you have the egg cell, with electric pulse basically merges them together and it starts dividing in the surrogate mother and forms the embryo. There's natural twinning and there's artificial embryo twinning. Egg and sperm, they both divide. This is done in a petri dish. They develop, and then you have two separate but identical embryos. And back here, the clone supposed to look like the somatic cell donor because that is what its nucleus is. And the risks of cloning has a high failure rate. Cloning causes problems during later development, like this goat here that died in China when they tried to clone it. Abnormal gene expression makes retarded clones, and telomeric differences. They noticed in the research that cloned animals have actually longer telomeres, which gives them longer lives. And Dolly the sheep, she was born 1996 to 2003. She was the very first successful clone from adult cells. You gotta remember that. And this is how it's done. From Scottish blackface, the cytoplasm donor and the nuclear donor, electric pulse, it becomes a blastocyst, put in the and it creates Dolly. And here's her taxidermized remains in the Smithsonian. And that's it.